Hello everyone, I am Dr. Sirajul Akfaisi. I welcome you in my YouTube channel. Today in this video, I am going to talk about some important questions related to bioorganic molecules. First question is, what is protein sequencing? Protein sequencing is a technique to determine the amino acid sequence of protein as well as which conformations the protein adopts and the extent to which it is complex with any non-peptide molecules. The second question is, what is N-terminal amino acid sequencing? This is very important question. N-terminal amino acid analysis, determining which amino acid forms the N-terminus of a peptide chain, is useful for two reasons: to add the ordering of individual peptide fragments sequences into a whole chain. You know, my friend, the first round of Edman degradation is often contaminated by impurities and therefore does not give an accurate determination of the N-terminal amino acid. A generalized method for N-terminal amino acid analysis follows: react the peptide with a reagent that will selectively label the terminal amino acid, hydrolyze the protein, and determine the amino acid by chromatography and comparison with standard. So, if your question is like that, what is the N-terminal amino acid sequence, in short question, you will write the answer like this. Now, the second question is, what is C-terminal amino acid sequencing? C-terminal amino acid analysis. The number of methods available for C-terminal amino acid analysis is much smaller than the number of available methods of N-terminal analysis. The most common method is to add carboxypeptidases to a solution of the protein, take sample at regular intervals, and determine the terminal amino acid by analyzing a plot of amino acid concentration against time. And this method will be very useful in case of polypeptides and protein block N-termini, and C-terminal sequencing would greatly help in verifying the primary structure of proteins predicted from DNA sequences and to detect any post-translational processing of gene products from known codon sequence. Now, the last questions. State Sanger's strategy for protein sequencing. You know, Sanger's strategy can be outlined as follows: determine what amino acids are present and their molar ratio. The second important point related to Sanger strategy: cleave the peptide into smaller fragments. Separate these fragments and determine the amino acid composition of the fragments. And third, identify the N-terminal and C-terminal amino acid in the original peptides and in each fragments. Then organize the information so that amino acid sequence of small fragments can be overlapped to reveal the full sequence. So, now the next question is, give the Sanger sequencing reactions. The reaction is carried out by mixing the peptide N1-fluoro-2,4-dinitrobenzene in the presence of a weak base such as sodium carbonate. In the first step, the base abstracts a proton from the terminal amine group to give a free amino function. The nucleophilic amino groups attack 1-fluoro-2,4-dinitrobenzene, displacing fluoride. You can see the scheme: 1-fluoro-2,4-dinitrobenzene structure, and then amino acid sequence, then formation of DNA with the amino acid sequence. Acid hydrolysis cleaves the amide bond of the 2,4-dinitrophenyl labeled peptide, giving the 2,4-dinitrophenyl labeled N-terminal amino acid and mixture of unlabeled amino acid. You can see separated. So my friends, I hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.